Hey guys, my name is Max and welcome to Simple Biology. In this video we're going to keep talking about photosynthesis, which we started in our last video. Now, like I mentioned in the last video, photosynthesis is incredibly important to life as we know it. And so that's why we're going to be studying photosynthesis, and that's why we're going to be going into more detail in this video. Now, in the light reactions, there's a functional unit called a photosystem, and that's what will actually do the work. A photosystem is basically a set of multi-protein complexes called the reaction center complex in the middle, and a set of light harvesting complexes all around it. And so each of these is made up of various pigments and proteins. In order to the proteins keep everything in place, and the pigments will be what capture the light and other energy in order to push an electron up to the primary electron acceptor, which will then be reduced. And so the whole point of this photosystem is in order to reduce that primary electron acceptor. This little yellow light represents the electron transfer between this special type of chlorophyll pigments to the primary electron acceptor. We'll be getting into all this a little bit more in just a minute. So these photosystems, like I said, are used in the light reactions. And there are actually two different pathways that electron can travel on from the photosystem. You have both linear and cyclic electron flow. I'm going to be going back and forth between this diagram and those words. Essentially, linear electron flow, this is that photosystem I just described. Linear electron flow, that electron will travel up, go down an electron transport chain, be pushed up one more time, before falling down to NADP plus reductase, which will produce NADPH. NADP plus is an electron acceptor that's used later on, and NADPH is used in the Calvin cycle in various functions. So we're going to be talking about linear electron flow first. And in linear electron flow, both of the photosystems are used. And in addition to that, both ATP and NADPH are produced. So the photosystems are actually named kind of backwards, photosystem 2 and then photosystem 1. They were actually named in the order they were discovered rather than the order that the electrons flow, which is confusing, but you'll get it, I'm sure, by the end of this video. In each of them, there's that special pair of chlorophyll molecules that I mentioned earlier. And in this one, it's called P680 plus, or P680 plus or just P680, depending on whether or not it has this electron. In the other one, it's P700 plus or P700, and that number corresponds to the ideal wavelength of that pair of chlorophyll molecules. The difference is just in a few protein interactions that change its specific properties. So, like I said, we're going to start with linear electron flow. And the electrons will actually come from a water molecule onto that P680 plus, reducing it to P680. And then by the action of all that light, which is kind of harnessed by all the different pigments, that electron will be pushed up to the first primary acceptor. Then it'll go through several proteins, such as cytochromes, in this electron transport chain, which functions very similar to the electron transport chain in respiration. And all of this is used to produce ATP. Then when the electron finally gets to the P700 plus in photosystem 1, all those pigments push it up to the second primary acceptor, where it'll fall down to NADP plus reductase through a little, this intermediate is called ferredoxin. Now on the other hand, we have cyclic electron flow. And in cyclic electron flow, only photosystem 1 is used, that second photosystem we showed. In addition to this, only ATP is produced rather than both ATP and NADPH, and no oxygen is produced, which is actually kind of interesting. In linear electron flow, that ferredoxin will actually allow its electrons to flow back into the electron transport chain rather than going down to the NADP plus reductase. And so as that electron travels through here, this light will actually be directly used to produce indirectly, but will only be used to produce ATP. And so by using cyclic electron flow, the cell in the chloroplast can produce as much ATP as it wants. Thus, the cell doesn't actually have to use respiration to break down the sugars formed in the citric acid cycle in order to get ATP. They can get it directly from cyclic electron flow. Now, after the light reactions, we of course have the Calvin cycle. Now, I've alluded to the Calvin cycle a few times throughout this video and the last, but now we're finally going to get into the details of how the Calvin cycle works and the various processes involved in it. Now, there are actually 3 phases to the Calvin cycle, which are known as carbon fixation, reduction, and RuBP regeneration, or ribulose biphosphate regeneration. First, we're going to go into carbon fixation. Now, carbon fixation is the part of the Calvin cycle that attaches carbon dioxide to ribulose biphosphate. Now, in this diagram, carbon fixation is just this kind of top portion. Ribulose biphosphate is a 5-carbon sugar, and after carbon dioxide is added to it, it has 6 carbons. The enzyme that catalyzes this reaction is known as rubisco. Now, rubisco is actually one of the most abundant proteins on the planet, which is actually really interesting to think about, and it shows how important photosynthesis is. But this 6-carbon sugar is very short-lived. In fact, it almost immediately splits into 2 phosphoglycerate molecules. One will continue in the Calvin cycle, and one will go on a different cycle. One will do the Calvin cycle somewhere else. But we're just going to follow one of those phosphoglycerates. And essentially, that phosphoglycerate will be the end of the carbon fixation phase, and we'll be getting into reduction. Now, reduction is the process that actually produces glyceraldehyde 3-phosphate, or G3P. Now, G3P is what's thought of as the product of the Calvin cycle. It's the molecule that's actually exported from the chloroplasts in order to form different molecules in other places of the cell. You can actually use 2 glyceraldehyde 3-phosphate molecules in order to produce glucose, which a lot of people think of as the product of photosynthesis. Glyceraldehyde 3-phosphate is a more accurate product, but it's kind of irrelevant. This is the reduction phase. Like I said, phosphoglycerate is used to produce glyceraldehyde 3-phosphate. Now, there's a series of intermediates that we're not going to get into, but essentially it uses 2 ATP and 2 NADPH. This NADPH will actually add electrons as well as hydrogen atoms to the 3-carbon sugars, and that's why this phase is known as reduction. When that sugar gains electrons, it's being reduced. Now, the last phase is RuBP regeneration, and this glyceraldehyde 3-phosphate, it can't actually advance in the Calvin cycle on its own. In order for it to get to this ribulose biphosphate, you have to take 4 more glyceraldehyde 3-phosphates and use all of them together and rearrange their carbon skeletons in order to form 3 ribulose biphosphate molecules. If you kind of count it up, do the math, you notice that there are actually no carbon dioxides gained or lost. This is merely a rearrangement of the different of their shapes and structures. This phase also uses ATP, but it only uses 1 ATP per ribulose biphosphate molecule. Finally, we're going to close by just accounting for all the different molecules that are used in the Calvin cycle. For every carbon dioxide that's fixed, you're going to use 3 ATP and 2 NADPH. Those come from over here where you use 2 of each, and here where you use 1 ATP. It's also important to realize that one turn of the Calvin cycle only actually fixes 1 carbon atom. In order to have a net production of a glyceraldehyde 3-phosphate molecule, you need to do 3 whole turns of the Calvin cycle. In order to have a net production of a glucose molecule, you would need to do 6 turns of the Calvin cycle. So that's important to know. That's really it for now. Photosynthesis is as simple as that.